Hello everyone, welcome to VLearn and welcome to the course Introduction to the Genetics. I am Ashish Kumar, your host for the Introduction to the Genetics course. Hope you enjoyed the last class. If you have any doubt at any point of time, let me know and I will try to answer every of your doubts. In last class, we studied about how Mendel did his experimentation part. So he did his experiment in 3 steps. The first step was selecting a parent plant. The second step was cross-pollination. And the third step was self-pollination. He did all these 3 steps for each of the character and obtained the same result for all these characters. Now in this class, based on the experimentation done by Mendel, he came with the postulates, or you can say principles of inheritance. So these are known as principles of inheritances. So there were some principle of inheritances discovered by Mendel by experimenting with Python sativum. So these principles are basically what we know as principle of inheritance. Basically, there are 5 principles of inheritances. The first principle is principle of paired factor. Principle of paired factor. The second principle is the principle of dominance. The third principle is principle of segregation. Fourth principle is principle of purity of gametes. And the fifth principle is principle of independent assortment. Principle of independent assortment. Out of these 5 principles, we have got 3 laws. So he gave 5 principles, and out of these 5 principles, 3 are law. So basically, if we talk about the laws, there are 3 laws. First is principle of dominance, which is first law, principle of segregation, second law, and principle of independent assortment, which is known as third law. So out of these are the first, second, and third laws. So these were the laws. So he gave 5 principles. But out of 5 principles, 3 are laws. We will discover each of these principles in next few classes. In today's section, we are actually going to cover principle of paired factor. So let's start with first principle, principle of paired factor. So first principle, principle of paired factor. After studying and experimenting for so many years, he came with principle of paired factors. These all principles are actually the result of the experiments conducted by Mendel for around 7 years. The first principle is principle of paired factor. The principle of paired factor says something was passed down. He didn't know what this something was, but he said something was passed down from one generation to next generation unchanged. So here the keywords are something and unchanged. So something is passed down unchanged from one generation to the next generation. At this time, when Mendel is experimenting with Poisson sativum and Mendel discovered these laws, there were nothing discovered like gene, chromosome, DNA, mRNA. He didn't even know the concept of gene, what a gene is. So he was totally unaware of these terms. So this something, he called this something which was passed unchanged from one generation to the next generation. He called this something as unit factors. He called this something as unit factor. The question which comes in NEET exam is, the unit factor term is equivalent to the gene. So genes are passed down from one generation to the next generation unchanged. So we will study about gene and then we will come back to the principle of paired factor. So what a gene is, the concept of gene. A gene is basically a segment of DNA. If we talk about gene, so gene is a segment of DNA. And this segment of DNA is basically present on the chromosome. If we talk about the structure of a chromosome is a rod-like structure. Chromosome is a rod-like structure on which is basically made up of DNA. And on this rod-like structure, small segments are present. These small segments are what we call gene. In human, there are approximately 25,000 genes. The number is not fixed, it can vary. So the number ranges from 25,000 to 30,000. Or we can say there are about 25,000 genes present in entire human genome. The number of genes on a single chromosome is also not fixed. All chromosomes have different number of genes. Like first chromosome have most number of genes in human. And these numbers are basically not fixed. The second is the position of gene. The position of gene, let's say this gene is basically present on this particular site. This particular site on which this gene is present on chromosome is what we called locus. So the site on which gene is present is what we called locus. So locus is basically the position of the gene on chromosome. So position of the gene on chromosome is what we called locus. Now from this word gene, we will study the concept of dominance and the concept of recessiveness. We will go and deep dive into the concept of dominance and recessiveness in this concept only. So how the genes are related to the dominance and recessive phenomena. Let us say this gene is an example of growth hormone producing, because basically all the hormones, most of the enzymes and hormone are basically made up of proteins. So this gene codes for growth hormone in plant, which is gibberellic acid. Let us say this gene undergoes transcription. What is transcription? Transcription is basically formation of RNA molecule. So from DNA, RNA is formed. This is what we call transcription. So this is the mRNA formed from this particular gene. And we have considered this gene as a growth hormone in plants, which is responsible for increasing the height, internodal elongation, which is basically help in internodal elongation, which in return help in tallness of the plant. So this mRNA, which is produced through the transcription, this mRNA undergoes translation. This process of translation helps mRNA to produce protein. If we talk about protein are one of the most abundant biomolecule in our body. And protein, basically if we talk about protein, almost all enzymes are basically protein. Almost all enzymes are protein. So if we talk about protein are enzymes. And you know that enzymes help in enzymatic reaction. So all the enzymatic reactions are basically carried out by these proteins, which are formed by translation through mRNA. Let us say this protein is a growth hormone, which is gibberellic acid in plant. The main function of this hormone is internodal elongation. This gibberellic acid helps in internodal elongation. This internodal elongation in turn will increase the height of the plant, right? So it will increase the height of the plant. So if we talk about this growth hormone, if it is undergoing normal rate of transcription or increased rate of transcription, let us say that increased rate of transcription, increased rate of translation, that means increased amount of gibberellic acid, that is protein, is formed, which will increase the internodal elongation. If this internodal elongation happens, the height of the plant is increased, and this is what we consider a dominant character. So we can say that dominant character is when the rate of transcription is high and when the rate of translation is low. So if the rate of transcription of a particular gene is high, that means that the gene is dominant, because that character is expressed, right? But if we talk about growth hormone in the other scenario, let us say this growth hormone undergoes some mutation. Mutation is a general term, all of you know. We will also discuss this term when we study the mutation separately, so don't worry about that. So mutation is a sudden change in your genetic or sudden change in your genome or DNA. Let us consider this part after undergoing mutation undergoes transcription and the process of translation. By the process of the transcription, the mRNA is formed. But this mRNA will not be similar to the mRNA in the first place, or in the first case we have studied. So this mRNA is basically the changed mRNA, or you can say the faulty mRNA. This faulty mRNA undergoes the process of translation and produces the less amount, because the translation will be affected. So the rate of transcription is lowered, rate of translation is lowered, and in turn the protein synthesis is lowered. So the amount of protein which is being produced, so protein synthesis will be decreased. Protein is decreased. What does this mean? This means basically we are going to have less amount of growth hormone, less amount of gibberellic acid means. And gibberellic acid is basically, responsible for increasing the internodal elongation in the plants, and in turn the height of the plant. If less amount of gibberellic acid is present, that means internodal elongation will not happen, or will happen in low amount. That means the height of the plant will be affected, and this height will be lowered. This character is said to be recessive character. We will continue this in next class. Thank you.